International Webinar Lecture. I would call Ms. Nitya Sri, final year biochemistry student, to deliver a welcome address. Over to Nitya Sri.
Yes, sir. Sir, am I audible, sir?
Yes, ma. Yes, ma. Yes, ma. You are audible.
Okay, sir. A dream written down with a date becomes a goal. A goal broken down into steps become a plan. A plan backed by action.
Nitya, please unmute your mic.
Sir, yes, sir. Shall I
You can mute.
start, sir? Okay. A dream written down with a date becomes a goal. A goal broken down into steps becomes a plan. A plan backed by action makes your dreams come true. A very good afternoon to one and all present here for the third session of the International Webinar Lecture Series. It's a moment of great privilege and honor for me to give a hearty welcome you all. I would like to express my sincere and warm welcome to our beloved President, Dr. M. Aruchami sir, who is an inventive person, guide the institution for the path of success. I would like to welcome the most dedicated, hardworking, for me to welcome our adored principal, Dr. M. Lakshmanaswamy sir, who stimulate and motivate us to achieve our goal. I'm glad to welcome our chief guest, Dr. Srinivasan Jayaraman sir, Assistant Professor in Biomedical Science, Faculty of Science, University Tunku Abdul Rahman, Kampar, Malaysia. So Dr. Srinivasan sir has obtained his PhD in biochemistry from University Tunku Abdul Rahman, UTAR, Malaysia. He has almost 16 years of professional teaching experience in higher education by educating multinational students from various disciplines of health and life science. His excellence in teaching and innovation in higher education has awarded him Vice Chancellor's Award for Innovative Teacher, Outstanding Teacher Award, and Teaching Innovation Award by INTI, Literate International Universities, Malaysia. And also, he was the finalist for the National Outstanding Education Award 2016 by Private Education Cooperative of Malaysia. Further, he received KSMCB Travel Grant Award in the International Conference of Korean Society of Molecular and Cellular Biology 2018 KSMCB held in Seoul, South Korea. He has shared his research finding in more than 20 international conference and research seminar at 5 different countries including Malaysia, India, Thailand, Indonesia and South Korea, and also published his research article in numerous Scopus and Web of Science indexed journals. Further, Dr. Srinivasan sir has been an invited speaker for a number of international conferences and seminars. He has organized a few international conferences and also involved as a scientific committee member for several international conferences. His major research area are toxic effect of zinc, silver, titanium dioxide nanoparticle on the nutrient microalgae, then environmental screening of metallic nanoparticle using microalgae as a bioindicator, impact of metallic nanoparticle on the nutritional property of microalgae supplement, antimicrobial property of zinc oxide nanoparticle on pathogenic bacteria and fungi causing skin and wound infections, effect of green synthesized zinc oxide nanoparticle using medicinal plant extract on skin dermal cells, and synergic effect of zinc oxide nanoparticle and medicinal plant derivatives on the cancer cell. And I today this session is going to be he shares his knowledge about the nanoparticle as the alternative antibacterial effect agent. Once again, I welcome you, sir. And It's my gleeful to welcome Dr. Selvi Ma'am, Head and Associate Professor, Department of Biochemistry, who is the guiding confidant of our college. Welcome you, ma'am. I would cordially welcome the Organizing Secretary, Dr. K. Nirupama ma'am, Assistant Professor, Department of Biochemistry, and warm welcome to Dr. R.T. Narendrakanan sir, Session Coordinator, Assistant Professor, Department of Biochemistry. Both of them led the webinar to a great exposure to the knowledge. Welcome you, sir. Welcome you, ma'am. I wholeheartedly welcome the faculty members, scholars, participants of other institution and our institution going to make today's session to a great extent. Once again, on behalf of our biochemistry department, I welcome you all. I hope all of you will enjoy the session. Have a good day. Thank you for joining us.
Thank you, Ms. Nitya, for this wonderful welcome address.
Thank you,
And
sir.
before I hand over the session to Dr. Srinivasan, I just wanted to mention just a few days back, Nature published an article which is very closely related to this topic, today's topic. That is that the Kiladi village. It was one of the best example for nanotechnology, carbon nanotubes that those people used in 2,500 years back. So this was recently found in our Indian Archaeological Society, and they have identified those people that used the carbon nanotubes in the pottery to prepare their food and all. So this is one of the best thing in our India. So we are very much forefront of scientific knowledge all over the world. Right. So in this small introduction, the paper it's available in Nature journal, one of the topmost journal in the world. The journal is recently published 5 days before. So with this small introduction, I would like to hand over the session to Professor Srinivasan Jayaraman from Tunku Abdul Rahman University, campus, Malaysia. Professor, over to Professor Srinivasan. Thank you. Sir, now you can take a session, sir. Srinivasan,
Okay.
sir.
Sir, am I audible?
Yes, sir. Yes, sir.
Sir, my slide appearing, sir, the screen?
Yes, sir. We can review your slide also.
Okay, sir. Okay, sir. Thanks for the warm welcome. And I would like to thank the management of Pongunadu Arts and Science College for the invitation to deliver talk in your web series. And I would like to convey my special thanks to Dr. R.T. Narendra Kannan for his invitation to deliver this talk. So actually, I'm going to the topic now. So today's topic, nanoparticles as an alternative antibacterial agent. So before going to the topic, I just want to introduce about our university. Our university is University Tunku Abdul Rahman University. It is in Malay here. Actually, it is ranked in top 500, 1 to 600 according to the Times Higher Education World University Ranking for 2021. And our university is number 2 in Malaysia after University of Malaya, which is ranked number 1 in Malaysia. So our campus is a very beautiful campus, which is located just below the Cameron Island. That's a very beautiful tourist location actually. And this is our faculty. Actually, you can explore about our university facilities, faculties through our university website. So the university website ID is here. Okay, so to our topic, nanotechnology as an alternative antibacterial agent. So we know nanotechnology is growing very widely, massively in recent days. Actually, the first growth is because of the industrial revolution. So we can see the nano-sized particles have been used mostly in all the industrial and the consumer products. So we are using the products made of nanoparticles in all areas of our daily life. Basically, what is nanoparticle? Nanoparticles are defined as the particles with at least one dimension size ranging from 1 to 100 nanometer. That's why it's called nanoparticle, because the measurement ranges in nanometers. So we can see here. This scale showing 100 nanometer. So the particle here, the diameter, when it is less than 100 nanometer in either one of the dimension, then we call this as nanoparticle. So nanoparticle, what is the specialty of the nanoparticle? Nanoparticle has small size. Because of small size, it has high surface area to mass ratio. And because of this high surface area and small size, they have high reactivity. That is their special physicochemical properties. So because of the special physicochemical properties, the nanoparticles are being used in applications ranging from electronic industry, communication industry, cosmetics, coatings, paints, packings, until the biological system. We are using in even biomedical science as drug delivering agents. So it has a wide range of applications. Now, why we need to think nanomaterials as an antimicrobial agent? So there are a few reasons here. So I put here because of rise in human population across the globe, especially the developing countries, with an increasing incidence of microbial infections. The population is increasing as well as the microbial infections also increasing. But available medications and therapies are insufficient to completely cure such microbial infections because of the development of resistance against various antibiotics. So this brings the risk of microbial infection and drug resistance. And now there is a demand for a new and compelling technology which has the ability to impede the existing problem. Now, when you talk about antibiotic resistance, this is a big challenge now among the health system. Previously, what happened? The antibiotic resistance happened only in the hospitals, strains. But what happened now? It happens everywhere. So because of globalization, excessive use of antibiotics in animal husbandry, aquaculture, and use of multi-broad spectrum antibiotic, these are the possible factors responsible for the spread of antibiotic resistance. So antibiotic resistance is a major critical issue because we can see here by 2050, there will be no effective antibiotic available if no drug is developed or discovered. That is the critical situation now. So we can see from this picture, by 2050, one person would die every day. In every 3 seconds, one person who died in every 3 seconds. You think of the complications. If one person can die in 3 seconds, in 1 minute, we can see 20 people. So think of 1 hour in 1 day. That is the problem now, critical issue. So that's why there is a need to search for alternative method for controlling the antibiotic resistant pathogens. So that's why we are looking for nanotechnology to overcome this problem. So now we are going to nanoparticles. So we have numerous nanoparticles. On that, particularly the metal nanoparticles have been proven to be antimicrobial agent. They act on broad range of target sites, not only extracellular and also intracellular compartment. That is a problem also. Many drugs act on extracellular part only, and those drugs are not able to enter into intracellular compartments so that they cannot kill the microorganisms which live inside the cells. But the nanoparticles, they can enter into the cells and kill the microorganisms which are located in the intracellular compartment. Because of the advantage of its smaller size, nano size, they are used. They have been already used in a number of materials. As I told you before, it is used in every applications. What we use every day in our house, mostly all from nanoparticles. They're all made of nanoparticles. So here, metallic nanoparticles have been proven to be effective for treating the infectious diseases, including antibiotic resistance. That's where they are coming now. So there is something good thing to feel that the metallic nanoparticles can be used to treat antibiotic resistance microorganisms. So here I show you the silver nanoparticles. That as silver nanoparticles have been proven to be an effective antimicrobial agent. Now here, so among the most commonly used metallic nanoparticles, zinc oxide, titanium oxide, silver. Zinc oxide, titanium dioxide, and silver are mostly used to control the microbial infections. However, to be on the good side, zinc oxide nanoparticle has greater advantages because of its highest photocatalytic efficiency and also the biocompatibility. When compared to the other nanoparticles, all the nanoparticles, metallic or non-metallic nanoparticles, zinc oxide nanoparticles have been proven to be least toxic to the human cells. That's why the zinc oxide nanoparticles are commonly used in skincare products. What are the skincare products we use today? All the body lotion, face cream, sunlight cream, all the sunscreen, all the creams are made of zinc oxide nanoparticles, because zinc oxide nanoparticles can fight against ultraviolet light, and also they can promote epithelial regeneration so that our skin cells will be looking healthier and shiny. That's why we think zinc oxide nanoparticles, since it's already being used in the skin lotions, all the thing, and it is proven to be least toxic to the human cells. So we try to focus more on utilizing zinc oxide nanoparticles in treating infections caused by microorganisms because of its least toxic effect to the human cells. And also, the zinc oxide nanoparticles have been reported to be effective on multi-drug resistant bacteria. So you can see beautiful pictures of zinc oxide nanoparticles here. So basically, what is the mechanism? How the nanoparticles, basically, they act and cause antimicrobial activity. So from this slide, you can see the first mechanism is production of reactive oxygen species, including superoxide, hydroxyl radicals, and hydrogen peroxide. You would have studied about the oxidative stress by reactive oxygen species. So what happened? Once the particle being exposed, once the microorganisms are in contact with nanoparticles, what they do, they will go and attach on the surface. The interaction of nanoparticles on the cell membrane leads to the production of reactive oxygen species. So we can see here, hydroxyl radicals and superoxide ions will act on the membrane and will cause membrane rupture, cell membrane rupture. Then following the cell membrane rupture, hydrogen peroxide can enter into the cell and cause damage to the cytoplasmic organelles macromolecules, including DNA. So then actually we can see, so what happened, all this reactive oxygen species causes destruction of cellular components, including DNA, proteins, lipids, and the cell membrane. So leading to loss of cell membrane integrity. So once cell membrane break, yes, the cytoplasmic organelles can be out, which leads to lysis of the cell. So another mechanism we can see here, once the particle come in contact with the membrane, it produces ROS, and also they can directly cause the particles, can directly cause the membrane damage on the bacterial surface, which will again lead to the influx of more nanoparticles and the zinc ions, causing more toxicity to the bacteria. Apart from this, the zinc ion, release of zinc, once we dissolve the zinc oxide nanoparticle in the medium, aqueous medium, because of the solution, the zinc ions will be removed, will be released from the nanoparticles, and they will cause disturbance in the enzymatic system, and thus cellular metabolism will be affected. That also will be leading to the cell death. So we can see the three major mechanisms: release of ROS, release of zinc ion, and direct contact of nanoparticles over the cell membrane. All these are the major three mechanisms leading to the death of the bacterial cells. So these are the three major mechanisms responsible for antimicrobial activity. Okay, so this is about the basic on zinc nanoparticles and how they act on microorganisms and killing them. Now I'm going to share some of the findings from my PhD study and also from my students' research projects. So we talk about ROS. Just want to show you what is ROS. We can see here, okay, this is from my study. So we used the fluorescent microscopy to demonstrate the ROS production. Yes. So from here, I used, actually in my research, I used Cyanobacterium, spirulina, and also Hematococcus. So we can see here, this one is spirulina, the filamentous one. The spherical one is Hematococcus. So what happened, when I expose the cells with the zinc oxide nanoparticle, we can see the green fluorescence. The green fluorescence indicates the presence of intracellular ROS. The middle one is the positive control, where the cells were treated with hydrogen peroxide. And the top one is a negative control, which appears in red color. The red fluorescence indicates absence of ROS, whereas the green fluorescence indicates presence of intracellular ROS, overproduction of intracellular reactive oxygen species. Just I want to show you, because I'm talking about ROS. I just want to show you the visual picture so that you can understand better. Okay, this is from my study. So now I'm going to share, because my study was slightly different, because I worked on microalgae and cyanobacteria. Now I'm going to share my students' work. This is my student work. That is, we tested the effect of zinc oxide nanoparticles on Streptococcus pyogenes. This is published in South African Journal of Chemical Engineering from Netherlands, which this belongs to Q1. So what actually we did? We tested the effects of zinc oxide nanoparticle on the Streptococcus pyogenes bacteria. So before we test the effect, we have to characterize the particle. So what we did scanning electron microscopy of the zinc oxide nanoparticle. In the scanning electron microscopy, we observe what is the shape and size of the particle. So we identified rod-shaped particles with the spherical particles. So we got rod as well as spherical particles, and we measured the size of the particles. So we got the different sizes: 52.6, 66.3, 49.1, and 31.4. So we took the average. The particle we used was 56 nanometer. So we used scanning electron microscopy to identify the shape and also the and also to measure the size of the particle. Number two, we use SEM EDUX, scanning electron microscopy with the EDUX spectrum. This is to test whether the particle is pure or not. From this spectrum, we can see it has got zinc and oxygen only, no other metal present here. So that we confirm what particle we use that contained only zinc and oxygen, not any other material. So EDUX usually used to confirm the elemental composition of the metals. Okay, so the bacteria, just I want to give you a brief introduction about this bacteria, Streptococcus pyogenes. It is basically a gram-positive cocci arranged in chains. This causes sore throat, tonsil inflammation. You might have come across throat infection, throat pain. Most of the time, the throat infection, the tonsil infection, tonsillitis is caused by Streptococcus pyogenes. And apart from the tonsillitis, actually this fever, sore throat, the tonsillitis, throat infection leads to heart valve damage, leading to rheumatic heart disease, where the heart walls will be damaged by the bacterial infection. This is a brief intro about this Streptococcus pyogenes bacteria, which is actually considered very, very pathogenic, very dangerous bacteria. You can see most of the people undergo heart valve replacement surgery. That is because they would have had this infection in their childhood. And the later stage, that completely affects the valve, and they have to go for valve replacement surgery. So one of the reasons for valve replacement surgery is this bacterial infection. Okay, so we just wanted to do the growth inhibition. How nanoparticle causes growth inhibition? Remember in the research, we have to always go with the low cost testing to high-fi test. It's always important. We always think we have to use high-fi instrument, we have to use high-fi methods to do the research. Not necessarily. We have to always start with a simple experiment which consumes very less cost. So what we do, we expose the bacteria in a suspension with a nanoparticle. It's called the turbidity method. After 24 hours of inhibition, we just want to see whether there is growth inhibition or not. You can see this is a negative control with no nanoparticle. And this one 10 ppm, 50, 100 positive control, they use vancomycin. We can see there is a decrease in the sediments of bacteria. So it somehow gives information that the bacterial growth has been inhibited by the use of nanoparticles. Next, what we did, okay, so using the turbidity, that means we measure the turbidity in spectrophotometer and then calculated the percentage of inhibition using this formula. Then we found significant inhibition. We found significant inhibition in bacterial growth from 10 ppm of zinc oxide nanoparticles after 24 hours of inhibition. We used one-way ANOVA to test the significance at the significant value of p less than 0.05. And actually also we plated on the agar plate to see any bacteria growing after the incubation. We can see here in the positive control purely no growth, and negative control you can see a lot of bacterial colonies. And in the nanoparticle 10, 50, 100, you can see the number of colonies are decreasing as the concentration of nanoparticles increased. So we can see in 0, 181 colonies, 10, 102, 50, 18, and 100, only 4 colonies. So it shows zinc oxide nanoparticle really killed the bacteria. It showed the bactericidal effect. So again, the colony, we calculated the growth inhibition percentage using the colony number, using the same formula. We want to do both the methods. The turbidity method shows bacteriostatic effect, whether the particle inhibit the growth of bacteria or not, but not necessarily it will show whether it killed or not. The colony count method on the plate only will show whether the bacteria has been killed or not. So that's why we use both turbidity method and colony method. Both the methods are very simple method. I mean, low cost consuming. Okay, next actually, as we know the mechanism, production of ROS, membrane damage. These are the major mechanisms for growth inhibition or death of the bacteria. So that's why we wanted to measure the amount of ROS produced in bacterial cells after exposed with the nanoparticles. So you can see here, even at 10 ppm showed significant increase in the production of reactive oxygen species. So you can see like 889%, 1000 plus, 1200, 1400% increase in production of intracellular reactive oxygen species. And remember, ROS always leads to lipid peroxidation. Lipid peroxidation, the ROS will cause peroxidation lipids present in the phospholipid layer. That's how it causes cell membrane rupture. So we also tested the lipid peroxidation level. This also showed significant increase even at the lowest concentration of zinc oxide nanoparticle used, 10 ppm. So basically, oxidative stress we assess by using ROS measurement and also lipid peroxidation. So we know ROS will cause damage to the bacterial macromolecules, DNA, and also the enzymes, so that the cellular metabolism mechanism will be inhibited. ROS also causes lipid peroxidation by interacting with the lipid layer and causing damage to the membrane, so that the bacteria will undergo lysis. So we can see actually we examine the morphological changes through scanning electron microscopy. So this is a control. You can see the cells are very clear and spherical, no structural abnormality. Whereas here you can see already the cells already destructed. You can see the structural changes, alterations in the bacterial cells. On this published article, you can see the membrane already damaged. You can see here it's already damaged. Here you can see there is a hole, membrane depression, membrane already gone. And here you can see the ruptured membrane debris. The cell already lysed. Only the membrane debris is there. Here also you can see invagulation in the membrane. So these are the, we already showed by scanning electron microscopy, the particle really caused damage on the cell membrane and cell structure, so that the bacteria can die. So that's what we demonstrated. And now there is another test called FTIR. FTIR is usually done. What was responsible for attaching the nanoparticle? Because we can see the particle got attached over the bacteria. After getting attachment only, the particles will damage the membrane, produce ROS, and then further enter into the bacterial cells. So we want to find out which actually helps in binding the nanoparticles of the cell of bacteria. So FTIR, it is called Fourier transformed infrared analysis. We found that the cell wall protein polysaccharides, the peptide molecules, the cell wall polysaccharides and proteins, they are the one involved in binding the nanoparticles over the surface of the, so we can see actually these are the functional groups involved: hydroxyl, amino groups, carboxy group. All these groups are involved in binding the nanoparticle over the surface of the bacteria. So once this nanoparticle go and bind over the surface of bacteria, which will cause imbalance in the membrane potential, leading to loss of membrane integrity and also imbalance in the transport system of the nutrients, and thus it affects respiration and energy process, leading to death of the microorganism. So that's why in our study we comprehensively demonstrated from the growth inhibition as well as oxidative stress, morphological damage, and also what functional groups were involved. What are the functional groups involved in binding the nanoparticle on the surface of bacteria? So then we published our paper in the South African Journal of Chemical Engineering. This is one student work. So next I go to the another student. Just want to share you a few things only. So this one, Pseudomonas aeruginosa, this is a gram-negative bacteria. This is again very dangerous bacteria. Most commonly, it undergo multiple drug resistance. So we can see it causes a lot of infections, mostly UTI, respiratory tract infection, skin infection, blood infection. It's bacteria, and it can cause systemic infection all over the body. In severe burns patients, cancer patients, and AIDS patients, those who are immunocompromised. You can see these are the few. I want to show you the infections caused by the Pseudomonas aeruginosa. You can see the green color because this bacteria produces a pigment, a green color pigment. So fine. Actually, this one again we can see here the negative control, massive growth of the bacterial colony. Positive control, no growth. We used tetracycline as a positive control, and the concentration 10, 50, 100. So the number of colonies decreased, indicating the bacterial cidal effect of zinc oxide nanoparticle. So again, this student did both colony count and also the turbidity method, and showed from 50 for this bacteria, 10 was not significant. So at 50, the significant growth inhibition was not reported. From 50, she reported 50 and 100 showed significant growth inhibition. Same thing, ROS showed ROS production significantly from 50 ppm. Lipid peroxidation also same. Okay. Okay, next actually we can see another student did again the growth inhibition test using the turbidity method and also the growth inhibition method. And you can see here the colony numbers also decreasing. Surface interaction we can see here is clearly you can see the structural damage caused by the zinc oxide nanoparticles. So it's clearly shown how the structure being destroyed. The FTIR showed the involvement of proteins, polysaccharides, similar to the previous studies, in binding the nanoparticle over the surface of bacteria. And apart from this bacterial studies, we worked on the cyanobacterium, spirulina, that also showed the zinc oxide nanoparticles significantly caused growth inhibition on cyanobacteria spirulina. So we published this paper in US-based journal, Q1 journal, and also we tested and showed the cytotoxic effect of zinc oxide nanoparticles and microalgal Hematococcus pluvialis. This paper also we published in Q1 US paper, PLJ, and we do have other studies we did on Chlorella vulgaris that also showed even short term, even 2 hours exposure to zinc oxide nanoparticles also showed significant inhibition of growth on Chlorella vulgaris. And also we showed the oxidative stress effects on Hematococcus pluvialis. So demonstrated upon exposing with the zinc oxide nanoparticles, the Hematococcus showed the oxidative stress effects. Actually, I have a lot of my students' project to show, but due to the time constraint, I just want to share only a few things with you. So you can actually ask more things if you want during the Q&A session. So with this, I'm finishing my presentation. We can have more interaction during our Q&A session. Okay, thank you, Asaf. Now.
Yeah, Professor, shall we go for question session?
Yes, sir. We can go.
So the participants you can can ask any questions if you have any clarifications or discussions. The session is open for all. Please unmute and you can talk instead of computer chatting.
Hello. Sir,
Hello.
good afternoon, sir.
Hi, good afternoon, madam.
Sir, actually, xenoxide nanoparticles or whatever it may be, either it is damaging only the cell membrane or it is creating the apoptosis on the cell. Means
Yeah, actually, they do cause apoptosis. They do cause apoptosis. So they cause cell membrane damage and also produce ROS. Upon ROS, they will undergo apoptosis. Yes, madam, they do undergo apoptosis. Actually, now my students are studying the apoptic effect on human cancer cells and also the normal cells. We are doing it now.
okay.
Yeah, they do cause. Thank you, madam.
Thank you, sir. Thank you.
So any more questions? Or any clarifications? Anything from the students, even, or any scholars, those who are working in nanomaterials? So I think, Professor, you can share your mail ID.
Okay,
so
okay,
that,
sir.
So anyone have any questions like that, they can discuss with you. I think it's better right now, right, sir?
For sure, sir. I will put my mail in the chat box. So actually, for the participants, actually, our university do have masters and mostly PhD programs, and we have. I mean, Dr. Narendra Khanna knows actually we have quite a range of facilities and instrumentation. So we can actually, sir, we already talked about it. So we can go for research collaboration, the student exchange. Your students can come over to our campus. Our students also can come over to your campus for some part of research work that we can do, sir. We are really looking for it.
So that we will work out. So we will work on it. Surely by 2021, we will sign the MOU.
Yes, sir.
After this pandemic situation, we will make it up. Yes, sir. And I sincerely express my sincere thanks to you, Professor, once you readily accepted my invitation, and also in your class timing as well as the busy schedule. It's my very pleasure to invite Dr. Venkatesh, Assistant Professor of Biochemistry, to deliver a vote of thanks. Over to Venkatesh.
Yes, sir. So it is audible, sir.
Yes, you can proceed.
Very good afternoon, one and all present here. It is my privilege to propose the vote of thanks on this valuable occasion of webinar series. Foremost, I convey my sincere gratitude to our Honorable President, Dr. M. Aruchami sir, who provide us an useful support and guidance. I extend my heartfelt gratitude to our beloved Secretary, Dr. C. A. Vasugi ma'am, for her continual motivation and a remarkable presence at all times. Thank you, madam. I also convey my sincere thanks to our college principal, Dr. M. Lakshmanaswamy sir, for his constant encouragement to conduct this program. I would extend my sincere gratitude to Dr. R. Shivaprakash, Associate Dean and R&D, and Dr. S. R. Madan Shankar, Dean Academics, who have contributed their immense support to this event. I convey my heartfelt thanks to Dr. V. Selvi ma'am, Associate Professor and Head of the Department of Biochemistry, who has given her full moral support to this webinar series. I would like to convey my wholehearted thanks to our resource person, Dr. Srinivasan Jayaraman, for his wonderful session. The topic, nanoparticles as an alternative antibacterial agent has really enlightened our young mind with new thoughts. Thank you, sir. I also want to thank the organizing secretary, Dr. K. Nirubama, and the session coordinator, Dr. R.T. Narendra Kannan, assistant professors of Department of Biochemistry. Finally, I am very grateful to the participants from our college and from various institutes for their active participation in this session. Once again, I thank you all for your kind attention. Thank you.
Thank you, Dr. Venkatesh, for your wonderful vote of thanks. Now I just mention you. I just invite for everyone for our next session. It will be on Saturday. So Saturday morning 9:30. The time may be the session. We will check it. College is opening on the day, so let us find out whether the 9:30 or 10 o'clock. So that on the Saturday we have a next session. It is so we have a 2 days break. So Dr. Jagadish Chandra Bose Rajendran is a postdoctoral researcher, biomedical medicine, Masonic Medical Research Institute. The topic will be derived vesicles, isolation, functionalization, and biomedical applications. It is on 5th of December, 9:30 a.m. At present, we will intimate you if any changes. So thank you, Professor Srinivasan, for your wonderful lecture. And thank you. I thank everyone for your patience and listening the lecture. I think somewhere you might have some point of idea about nanoparticles, since we have recently we have exposed to the world that we are pioneer in nanoparticle 2,500 years before, based on the KLD research published in Nature journal. So it will be more exponential research will be in the nanoparticles. So thank you, Professor, for your wonderful session. Have a good evening to you, and good afternoon to our Indian participants. Thank you so much.